A very good morning to one and all. Welcome to the second session of uh, our international webinar on occasion of National Nutrition Month. As all of us know that micronutrient deficiencies are one of the most prevalent issues that our country is facing, and our government as well as all our nutrition bodies are working towards eradicating this issue. Also, um, preconceptional nutrition lays the foundation for health and longevity of a person's life. That's why we have two very eminent speakers to talk to us and give us more information and latest research on these two topics in our morning session today. I hereby welcome Dr. Veena Yadi to welcome our first speaker for the morning. Over to you, Yadi, ma'am.
Uh, Very good morning and thank you, Dr. Neha. I feel honored to introduce a very well-known scientist who is also a reputed teacher, guide, and a mentor, and Dr. Madhavan Nair. Dr. Madhavan Nair has done a lot of work in the area of micronutrient deficiencies, bioavailability, food fortification, and today he is going to talk about food synergies, evidence-based dietary approach to address micronutrient deficiencies. I'm sure, like me, you are also very eager to listen to him. So I will quickly go through his CV. Dr. Nair has done he is a fellow of 3 national academies and member of a large number of national and international expert committees on public health nutrition. He has done his post-graduation in biochemistry from Kerala University. He has done his PhD in biochemistry from Osmania University, and he worked at the ICMR NIN for his research experience. He has done his postdoctoral training as a Fogarty International Fellow, very prestigious Fogarty International Fellow for Advanced Study in Health Sciences, NIH US, at the International Center for Control of Nutritional Anemia. Dr. Nair has worked for more than 36 years at the ICMR NIN and developed micronutrient bioavailability as a focus area. His expertise includes translational research, public health nutrition, and micronutrients. He coordinated the expert group of the ICMR to revise the nutrient requirements and RDA for Indians in 2009-10 and in developing the national standards. He has more than 150 papers to his credits. He has published papers and book chapters. He has guided many students for PhD in biochemistry as well as in nutrition. He has received many awards, to name few: the Public Health Service International Research Fellowship Award of the Fogarty International Center for Advanced Study in Health Sciences, ICMR BGRC Silver Jubilee Oration Award, Fellow of the National Academy of Medical Sciences and National Academy of Agricultural Sciences. He was also founding fellow of the Telangana Academy of Sciences. Then he has also received Dr. Srimati Mrunalini Devi Puar Memorial Oration, from Foods Nutrition Alumni Association of MS University. He has also received Dr. S.G. Srikantia Memorial Lecture Award. Next slide. Now, Sir is also chairperson of scientific panel on labeling and claims advertisement of FSSAI, and he is member of so many expert committees. You know, I'm just going through briefly because you will know the amount of expertise he has in various fields. So I'll just read out quickly. Task Force Committee on Biotechnology-Based Program for Rural Development and SC/ST Population, DBT, Government of India, New Delhi. Technical Advisory Group, UNICEF, CNSS, MOH, FW, GOI, New Delhi. Scientific Panel, Nutrition and Fortification, FSSAI, GOI, New Delhi. Expert Committee, Approval of Non-Specified Food and Food Ingredients, FSSAI, GOI. Expert Committee on Advertising and Claims, FSSAI. Project Advisory Committee, NABI, DBT, Mohali, Punjab. Expert Committee, Public Health Nutrition, DBT. Review Committee, Development and Transfer of Technology for Biofortification and Disease Resistance in Banana, BIRAC, New Delhi. Scientific Advisory Committee, National Institute of Plant Genome Research, DBT, Expert Group, CSIR, Nutraceutical Mission, CSIR, GOI, New Delhi, International Scientific Committee, Choices International Foundation, Netherlands, the Data Monitoring Committee, ICMR, Task Force Study on Childhood and Adolescent Anemia, Expert Committee, Revision of Indian RDA and Dietary Guidelines for Indians, ICMR, National Committee for International Union of Nutritional Sciences, INSA, INSA. Now, so it's such an impressive CV, sir. I don't know. I mean, you have done so many things, and I'm sure all will agree with me that we are extremely fortunate to have Dr. Nair with us today, who's going to talk about food synergies, evidence-based dietary approach to address micronutrient deficiencies. Very, very interesting topic. So over to you, Dr. Nair, to take over the platform. Thank you so much. Dr. Nair?
Sir, you are on mute.
Okay, okay. Can you hear me now?
Yes, yes.
Hello.
Yes, sir. Yes, sir, we can hear you. Sir, can we request you to put on your camera also?
It's off.
Yes, sir.
Okay.
Thank you, sir.
Yeah, good morning to all. And thank you, Dr. Veena, for that introduction, nice introduction you gave. And I am indeed very happy to be part of this meeting. That is, it's a National Nutrition Month. Earlier, it used to be only a National Nutrition Week. Now, it has been made to month. I think it's a good idea to have this sort of translational research and related messages being given to public through students. They are the medium for us to translate all these information to general public. And I'm also happy to be part of this international webinar organized by NSI Mumbai, and I wish all of you a very happy, fruitful nutrition month. I hope you will all do justice to the month, and wish all the very best. Let me go through my slide. I can operate it.
Yes, sir, you can change the slides yourself.
Okay. Yeah, I should probably start my lecture by saluting my institute, which is nurtured me for more than 36 years. I owe a lot to this institute, and whatever I am is because of this institute. And my mentor, late Dr. Narsingh Rao, who is the Iron Man, who introduced me to the world of iron nutrition. And whatever little I learned and going to present today is contributions from many of my own mentors, including Dr. Bhamji, she gave an excellent lecture yesterday, keynote address, and Dr. Shivakumar. Then, of course, there are so many directors, and also to my students, because I learned a lot from my own students because they taught me the essence of nutrition. I basically was a biochemist with very little knowledge, but interacting with these young minds, I could learn a lot of it. And the whole exercise has been very fruitful over the years, and whatever I'm going to present is their efforts and efforts of many others in the institute and elsewhere. This is the basic concept I am going to present. Of course, yesterday also there was a discussion balanced diet. I mean, we have to go on drilling this into the minds of people, what is balanced diet, because it has to be a persistent and education on balanced diet, we tend to forget about what is balanced diet, and we became more of taking monotonous diet. So I think this has to be stressed here. Of course, micronutrient deficiency, because the whole topic is to address micronutrient deficiency, and the key concept of what is food synergy, and how we can use that as a strategy to address the deficiency of these three micronutrients: iron, vitamin A, and vitamin D. And so all the whatever research I could obtain from literature and my own research, I will be presenting that as the evidence base, so that probably it could be incorporated into the national health strategy, and of course you can practice it and also disseminate the information. Balanced diet, in very simple terms, it has to be obtained from varied foods, that is a diversified food in quantities and proportions that need for energy, that the protein and minerals and vitamins. So these four important nutrients is the essence of what we talk about nutrition. In simple terms, not only we meet their requirements, actually, in fact, we make a provision for little extra, so that we withstand during the leanness. So when you don't get certain foods, this will take care of, like, certain nutrients are stored in the body, they will be used. So the key message for balanced diet is eat regular, healthy, balanced meal, so that we get all the nutrients all the time. And the essence of balanced nutrition is captured in the dietary guidelines of Indians, and that has been generated from the two national documents, that is the nutrient requirements and dietary allowance of Indians, and also the Indian food composition table. And it is synthesized, and that has been put into the dietary guidelines based on various studies. So we have the country specific guidelines, and that forms the scientific basis for balanced diet. And the key to this balanced diet is nothing but dietary diversification. And we will understand it little more when you go through the other PowerPoints. And now we know that unhealthy diet is a major risk factor for all communicable diseases. Yesterday, when Dr. Bamji also was highlighting about the issue about unhealthy diet and NCDs, non-communicable disease, and of course, the rapid developmental transition, economic transition has taken place, but that has not actually reflected in nutritional aspects. And also the urbanization affecting both consumption pattern and also the energy expenditure. All these put together actually increasing the risk factor for non-communicable disease. Also, there's a new term, the economic transition, epidemiological transition level, which is nothing but there are certain states which are epidemiological transition is at a high level, like states like Kerala, compared to states like Bihar, they are at the lower level, and the states those are at high level is states like Kerala, Goa, and other states. So there is actually the incidence of non-communicable disease has gone up in those states where the epidemiological transition has taken place. But in states like Bihar, there is no transition. Even now we talk about communicable disease as the major problem, though they have also emerging into the pandemic of non-communicable disease, the obesity and related things. And we could feel that we are all oversimplifying our diet. I think we also practice that because of convenience and various other things, but that is what probably we should not be doing, oversimplification of diet, which is very, very dangerous, and leading to no diversification of food, and that becomes the real cause of all non-communicable disease related to the food factors. And so we need to remind people to constantly eat well. And I think this forum, the NSI Mumbai and the National Nutrition Week or month, is I thought it's a good forum to repeatedly tell people to remind them that you eat well and do use a balanced diet. And the details of messaging. I mean, this is very important because the whole aspect of eating balanced diet depends on how well we diversify our diet. So we need to actually message people that we should eat at least a minimum of 3 balanced meals per day. I mean, like we know that a breakfast, lunch, and supper, and also eat at least. Okay, I mean, if you see the dietary guidelines, they have, it is, one portion of GLV is 100 grams, then 2 portions of other vegetables is 200 grams, and 1 portion of fruits is 100 grams. The total, it's almost like 400 grams of these fruits and vegetables together, one has to, this has to consume. This is applicable for an individual who is above the age of 7 years and, even beyond your 60 or 65 years. And individuals below that, the school age or preschoolers, it is comparatively less, but it's a large quantity of fruits and vegetable one has to consume. That is the essence of actually the balanced diet. And I have incorporated the next message that eat your fruits with all meals. I think that messaging is really good to get that synergy. I'll discuss about some synergy which is proved beyond doubt that they help in absorption of certain nutrients. Then, I mean, follow the national guidelines of all the aspects related to healthy lifestyle, like outdoor physical activity. This increases your, vitamin D. Also, the outdoor physical activity rather than doing an indoor physical activity. Now, dietary diversity, it's a in simple terms, we have divided it into 4 food groups. So it is defined as number of different food groups or foods consumed over a given reference period. So you have the energy providing foods, protein providing foods, and also the vitamins and minerals. Though the cereals also provide vitamins and minerals, but the major portion of vitamins and minerals comes from fruits and vegetables. And these are the simple classification of food groups. Now, the one thing you must understand is within the food group also you have so many variations you can make it. And that variety within the food group and across the food groups gives lot of benefits of both known and unknown nutrients. So the unknown nutrients comes only when because we know that there are, we talk about only 13 vitamins and some 15 minerals, but I'm sure there are much more than that because food as such is a complex matrix. The components are many, which are even unidentified. So the goodness of all this comes only when you have a variety of foods from each food groups. That is the diversity we must think of and practice. And I mean, nature is given us enough of it, and we should be actually, taking it as much as possible. It is an intelligent way of probably practicing all these things. Why diversify? I think I have given enough of messaging why we should diversify. Most of the vitamins and minerals comes from plant foods. They are the best source. Certain animal foods provide certain like vitamin B12 is one, then some vitamin A, vitamin D. These things also comes from animal, but most of the other vitamins, fats, vitamins comes from plant foods. And there are a lot of evidence, though moderate evidence, indicate that intake of vegetables and fruits is associated with reduced risk of cardiovascular disease, including heart attack and stroke, and including stunting. Because there is a meta-analysis showing that with increasing the number of food items or food groups, you could show a reduction in stunting. So we have been trying to test each nutrient in isolation and seeing whether they can impact stunting. But I mean, at least the research has shown that you need a complex food matrix probably to reduce stunting. And also, it diversification also gives us, or it establishes the food synergy and also the nutrient synergy for optimum absorption of all nutrients. That medium, that milieu, intestinal milieu, which sometimes, there are certain co-nutrients, they help in increasing the availability and absorption of certain nutrients. The best example, what I am going to discuss is the iron and ascorbic acid. Okay, so such nutrients are, their proximity at the site of absorption is very essential. And for certain nutrients, it should be available at the digestion site itself for it to get keep it in the absorbable form to be available for absorptive surface. Then this creates the ideal milieu for food and nutrient synergy, the co-nutrients. And of course, this strategy or this concept will take it for addressing micronutrient deficiency, especially for the three nutrients what I have already told you. Now, recently this has been published, the Comprehensive National Nutrition Survey. I think all of you should have this with you. It's freely available in the website, and you should go through it, understand the magnitude of various deficiencies in the country, and try to address them whenever you can. So I'm going to show the prevalence of the three important nutrients: the iron, vitamin A, and vitamin D. Of course, zinc also is there, but I'm not going to talk about that. But you can see that this is a little different from the NFHS database, which is relatively higher than this, because there is a difference in terms of the methodology adopted here for estimating the hemoglobin. But the major thing is, even though it is less, but the overall prevalence is, you can see it varies from 22 to around 40% in various groups, and the most vulnerable segments is the under 5, and of course you have the female adolescents. The other group, this is under 19 only has been done. The other survey is not yet over. So the problem of anemia is a significant problem, and also it has been well established that iron deficiency is the major cause of anemia in our country. Vitamin A is the other one. Though we have the National Vitamin A Prophylaxis Program, massive dose program, still, you can see this is based on the plasma vitamin A levels, which shows about 20% of the group under 19, population under the age of 19 is deficient. Similarly, vitamin D, which is an emerging problem because of the lifestyle changes and also pollution. There are various concepts, but the important thing to notice is that it is an emerging problem, and we need to tackle these issues. Now, what are the reasons? One is, of course, I say that there is no diversity. That is, our intake of these nutrients are very, very low in content. And that means unless you diversify like a balanced diet, you never get what you want to satisfy your requirement. So it has to be balanced, or you should find means of increasing the content. And the other one is, for certain nutrients, absorption is an issue. Though they are present, because of the dietary factors like the inhibitors, they will reduce the absorption and thereby reduce the available iron into the body. Like for iron phytate and polyphenol, for iron and zinc, these two are inhibitors, which is present in most of the food matrix. So it is very difficult to remove it or eliminate it. So and they have their own benefit. So it is probably the strategy should be, with all these things, how we can actually increase the bioavailability rather than eliminating it. So they are anti-carcinogenic, most of the phytates and polyphenols. And what are the options of preventing micronutrient deficiency? Of course, we know One is to increase, because low content, you need to increase the content. There are certain situations where we give supplementation of nutrients in large doses, prophylactic doses, or even therapeutic doses of given. But dietarily, if you want to increase the intake, the best one is the medium-term strategy, the fortification. Fortification is a deliberate addition of this micronutrient to a food vehicle, so that it reaches the entire population. So now the FSS has made a regulation to fortify wheat, rice, and also milk and oil. The wheat and rice for minerals or vitamins, but with respect to oil and milk, the nutrients what is suggested is vitamin A and vitamin D. And the third strategy is long-term strategy is nothing but the dietary diversification. And now for me, I mean that has to synergize even with the other two short-term and medium-term strategy. Then only it will work. Otherwise, it is going to be very difficult to correct the bring down the prevalence at a faster pace. It may take much more time to bring down the prevalence of this deficiency. But if you consider diversification as part of the main strategy, along with the short term and medium, then I think they should go concurrently. And we should understand that medium term should work with the long term, or there should be some system to increase the bioavailability by adding either food component that enhances the absorption, and thereby you actually reduce the time gap for reducing the prevalence. Also, the public health approaches like wash, water, sanitation, hygiene. So that is also is part of it. I think the Swachh Bharat Abhiyaan is one of the important one, probably may have some impact on reduction of these micronutrient deficiencies. Of course, the other one is promoters, increasing the promoters in the food what we take, so that increases the promotes the absorption, and reducing the inhibitors. I mean that normally is it's a difficult one as a food component, but there are guidelines of like avoiding tea and the food item that contains inhibitors should be avoided during a meal time. Okay, so tea should be avoided when you have your meal, so that it won't inhibit the absorption of certain minerals. Now, I want to just elaborate on the food synergy. I mean, the concept is to attain a balanced nutrition in a plant-based diet, so that the whole meal will become a conducive medium for better absorption of many nutrients. And it is defined as an additive or more than additive influence of certain dietary patterns, like the Mediterranean diets. They have a pattern. I mean, that pattern, if you follow, there is a possibility foods or food constituents on health. Food constituents could be even adding internet connections. Adding certain constituents like it could be a vitamin C or certain amino acids which increases the absorption. Now, in the context of micronutrients, we talk about food synergy or nutrient synergies in terms of enhancing the absorption or the bioavailability. So the advantage of this approach is it is very close to what we practice, that is population psych. So there is probably it is more acceptable for population that they would like to change and make an attempt to practice this to increase the availability of most of the nutrients. Only thing is it requires constant effort to create it as a national mission to address micronutrients, especially for iron. I think it has to be included as part of the policy or national mission. Now the focus is on these three nutrients. Of course, there is one I mean, India has now declared, or we have a mission or vision of Anemia Mukt Bharat. And the goal is to reduce anemia by 18% by 2022. And the rate of decline is at the rate of 3% points every year. Now, that means, 58 in the prevalence of children in anemia in children is 58%, and to reduce to 40%. That is about 18%. Like that, in all age groups, it is supposed to reduce 18% and make it to an acceptable limit, so that this target has to be met by 2022. Now, the major issue for iron is its bioavailability. Now, various studies from Dr. Narsingh Rao's group and our group and from St. John's, most of it shows the absorption is very, very poor. That also it is certain food groups actually has a lot of inhibitors. For example, millet. Yesterday, Dr. Bamji was talking about millet, but there is a problem of these inhibitors, the large quantities of inhibitors inhibiting mineral absorption. That's a well established. We can see here it's only 4 points as compared to the other. The food matrix has a influence that is mainly because of presence of the inhibitors. And other studies also show that the absorption varies from 5 to 10%, and that is the factor we have used to even derive the RDA for iron. Now, India is a country where the RDA for iron is the highest. For example, for adult male, the current RDA is 17%, 17 milligrams. Now, when you take in a Western country, it's only 6 or 8 milligrams. It's almost half of it. That's mainly because of the bioavailability. So their diet is more bioavailable because it's diversified. They have more of ascorbic acid containing foods, or even protein foods or the meat. They all increases the absorption. Or more processed foods also actually probably conducive for absorption. So this is a major issue. And when you talk about iron, I mean, we have to understand how iron is first, the three phases of iron absorption. The stomach or the gastric phase, then there is a intestinal luminal phase or the duodenal phase where the absorption takes place, and you have the post-absorptive phase. Now, the food actually, the food iron is in the ferric form, most of it, and it gets digested in the stomach. And one important attribute that is required for this type of reaction to take place is the pH of stomach or the gastric juice. The pH is 1.2 acidic pH. That's very essential for iron solubility. So if iron solubility is an important factor for subsequent absorptive process, it has to be soluble, and the pH is very, very important contributor for this. Now, all the reactions with respect to iron absorption has to take place at this pH, because subsequently what you see is the pH is going to increase to pH 7. It becomes more of alkaline pH because of pancreatic juice. And iron doesn't like this pH at all. Most of the iron gets precipitated as ferric hydroxide at this pH, or it will either chelate with the inhibitors, the polyphenols or phytates at the gastric pH, and it will be unavailable for absorption. Now, the only way to keep iron in the soluble at alkaline pH is to keep iron interact with absorption promoters like ascorbic acid, vitamin C, or the other organic acids, so that the complex which is forms at this pH is soluble even at alkaline pH. Now, that is very important concept that we are going to now test it using food matrix and food synergy, whether that that process takes place, and you can get an increased absorption. You can double the absorption or triple the absorption by that process of creating the ideal condition for iron to react with a medium or a food component that keeps iron soluble at even at alkaline pH. That's a simple nutrient synergy or food synergy we are talking about. Now, but look at the pH of gastric pH of Indians compared to the Western population. I think there is a pH difference of around 1. It could be because of the food habits, because the literature shows that the protein foods or the foods which contains lot of amino acids, the pH is higher. So you can see this, which is a carbohydrate and fat meal. Typically, our meals are like this. So where the acid production is very low compared to a 1 gram, this is actually 20 gram albumin, and this is 1 gram amino acid, and this is 20 gram amino acid. So amino acid means a complex or amino acid mixture. So amino acids are known to increase the availability of acid in the gastric pH. So ultimately, the dietary iron from our habitual diet, what is solubilized and what reaches the duodenum and what is absorbed is almost 5 to 10% if you are creating a conducive environment. Otherwise, it will be very, very, very poor. It will be only 1 to 2%. So that is the real problem for us to tackle anemia or iron deficiency anemia. Yeah. Now, this is with respect to food iron. Food iron, of course, it's in the non-heme. We are talking about only non-heme iron, not the heme iron, which is present in muscle and tissues related to that. Liver mainly contain non-heme iron because most of the iron present in liver is in the ferritin. So this is a ferritin molecule, but it is from plants. Plant also has ferritin molecule. It's known as phytoferritin. They can be isolated from grains. Now I will show you the studies we have done with the phytoferritin and how they behave at that different pHs and how it interact with the inhibitors and promoters, because that is very important message for us to practice. Whether is it similar to the elemental iron present in the tablets or the chemical sources we use, or is it differ in its absorption? But our studies shows that they are similar, because the ferritin actually has a core. It's like a football like structure inside, and gets accumulated in the complex form as hydroxides or phosphates. And this strategy of ferritin, incorporation of ferritin into these different grains, different crops, is used as a strategy for biofortification also. Like soybean ferritin into other rice or wheat, or now we have a pearl millet which is rich in iron because they have more storage ability through phytoferritin. Now this is a study we have carried out to understand this stability of ferritin, whether ferritin as such retains its structure, it escapes the gastric phase and reaches the duodenal phase is one thing we tested. What we have shown here is these are two different pH. This is phytoferritin isolated from green peas, because they are rich in iron. So we could easily isolate that and do the these experiments simulating the gastric phase and the duodenal phase and looking at whether they retain its integrity as ferritin molecule. You can see at pH 2, the ferritin actually dissociates and releases its iron compared to at pH 7. So, but the normal digestive process, it reaches gastric phase, then only it reaches the duodenal phase. So most of the ferritin ion is released at the gastric phase, and so it is behaving like the normal form of iron, the iron present in chemical forms of iron, like ferrous sulfate, for example. Now we tested whether when you incubate this phytoferritin with uh, ascorbic acid or with phytic acid, how actually it behaves in terms of its bioavailability. This is done in a cell line, human intestinal cell line, to see whether they actually have a better absorption or not. This is isolated ferritin is been subjected to these treatments. You can see, I mean, it behaves similar to the ferrous sulfate in terms of the increase in iron absorption, and also the reduction in absorption when you incubate with phytic acid. So the behavior of food iron and that of the chemical sources ferrous sulfate is very similar. And that means it goes through the same absorptive pathway. So you don't have to really worry about the chemical source versus the natural source. They are all same except heme iron. Now, so we thought we could address this issue by the food synergy, or how we can increase the absorption. Now, so we have to do a study to understand whether inclusion of this in the daily meal, because we believe that the transit time for iron, that is from gastric phase to the other phase, and the transit time for ascorbic acid, if you are given as a chemical, will be very different. That's why we used a food matrix that actually retards the release of ascorbic acid, and it may synchronize with the release of iron from food. So these two should synchronize, then only that synergy will be created for its absorption. Now this study was done using stable isotopes, because we had a Indo-US collaborative project with the University of Texas, Houston. So we could do this here both because it's easy for us to administer isotopes. So these are stable isotopes. It's not a radioisotope, so it can be administered, and it's very safe, like a normal form of iron. Any normal iron, what you take in the food or the tablet form, they all contain these as a stable isotope, the Fe57 and 58. These are the two isotopes we used for monitoring the or tracing the food iron. So this was done in a rice-based meal, and it was done in a residential school where they get a regular ration, and they are all standard meals. So it's very easy for us to take that platform and test it. We used both the females, girls, residential hostel, and that of boys. So two different adolescent groups, because adolescents, we know the group which require highest amount of iron. So, and we thought probably they will have better absorption, and it's a good platform to test it. So here, this is the typical meal they've been served, and we have, from our nutritive value of Indian foods, we found that Guava is the best source which can provide ascorbic acid. It gives 200 milligrams of ascorbic acid per 100 grams. So if you give 100 grams with this, which easily you can consume with food, so the one piece, it will be almost one fourth of this will be around 100 grams. Now the study is actually using cross our study design with the washout period and using stable isotope. These are the two isotopes we use, 57 and 58. And of course, I'm not going to talk about zinc, but we administered zinc also, because in same diet we could use both. So we got about 16 subjects after using all the selection criteria, and half of it, crossover means half will receive one diet, other half will get the other diet, and after the washout period they get interchanged. Now this is the meal. There are standard meals. I mean, most of them get around 450 grams per day, and this is the regular diet, and this is the modified. The only modification we did is introducing 100 grams of Guava in the meal. So they should consume as part of the meal. which actually provided around 180 or around 200 milligrams of ascorbic acid. So no other change other than introducing the ascorbic acid through our Guava. Now the results is very clear actually. You can see the absorption with regular meal is 9.7. This is in girls and this is in boys. It's a 8.6, and the modified diet, that incorporation of that 100 grams Guava has actually almost double the absorption of iron. You can see it's around 24%, and here it's around 20%. So the difference is almost around 11 or 12% increase in the absorption. So just by modifying the diet, you could double the absorption. That is actually an important aspect that we need to consider when you want to address iron deficiency or improving the bioavailability. Now that is only a single meal. Now, how do we actually show that if you take every day Guava with your meal, will it actually increase the absorption? Now here, so we have actually looking for a platform, and we found that the ICDS platform is the best suited for that, because they get a supplementary nutrition meal every day, and a lunch noon meal program is there, and this is the standard rice based meal. They have different packages depending upon the region, but in this region Telangana, it's the rice based, and they get this ration. And what we have done is, with this we actually included 25 grams of Guava. I mean, it's only 1/4 of what we gave to adults, because the quantity of iron, it's based on the molar ratio. And this is a a cluster randomized controlled trial. So we had about 16 villages, and then we randomized into, we had 3 groups. The first group is Guava group, then a control group, we had a banana group, because banana is low in iron, and third group we wanted to have a group without any fruit. But when we discussed with the mothers, they said they should also be given something. So we incorporated cucumber, because again they also have very low ascorbic acid, and we had given them, the intervention was for a period of 140 days, and we collected blood at baseline and also at end line. And end line we have more than 100, which is statistically sufficient sample for finding out the impact of intervention on iron status. Though we did other functional parameters like cognitive function and growth and development, and also we looked at the gut health, looking at some of the microorganisms that probably benefit because of presence of these fruits. It's very clear from this. You have the 3 bars. The first one is actually the bar showing the improvement in hemoglobin in Guava group, and the other 2, which is to the minus side, is actually the other group, which actually banana group and cucumber group. This is hemoglobin, this is ferritin. You can see this also increased, and also we have used serum transferrin receptor also as another indicator. That also showed an impact which is in tune with what we found for hemoglobin and ferritin in Guava group and the other group. So it clearly shows it's an intervention that actually enhances, improves hemoglobin, and also it actually builds iron store. That's what actually the meaning of increased ferritin. And also the impact on iron deficiency. You can see it actually reduced iron deficiency by almost 50%. The other 2, it's almost remains same. we measured the ascorbic acid level because we are giving every day vitamin C through Guava. Definitely, there should be an impact on vitamin C. Yes, it showed an increase in ascorbic acid in serum around that's in the Guava group, and also, I mean, we could find a significant difference in folate also because Guava has a little higher amount of folic acid compared to the other two food matrices. So this clearly shows providing preschool children vitamin C rich Guava as a fruit, as a complement to the cereal based meal increases hemoglobin, it reduces iron deficiency, and also, of course, we have got only an impact on upper respiratory infection, the symptoms related to that.
Sir, sorry to disturb you. Your session is going great, but now we have only limited time.
Yeah, I will finish it. I will finish it. Definitely, I will.
Thank you, sir.
Yeah. This is, I mean, we have done only with Govas, but there is literature to show that papaya also can do the same job. Here, this is a maize meal with they have first tested with ascorbic acid 70 milligrams, then maize with 150 grams of papaya, which provides 66 milligram of Guava, and this is maize without any of these things, and you can see clearly the increase in absorption. With the papaya, it's around equal to that of ascorbic acid, and also much more than what you find with this one. Okay, so if Guava is not available, papaya can be used. And also we actually looked at literature to see what are the other sources that has been used as a matrix for vitamin C. Mango, which is less, but it is doubling the absorption. Banana is only less, but the others are not useful for increasing the absorption of this one. So I have actually tried to compile the natural sources of iron absorption promoters. This is the ascorbic acid rich fruits and vegetables, which could be replaced with Guava. You can use as a alternative. The only thing I wanted to inform you that the gooseberry, which is actually high in ascorbic acid, but it is rich in polyphenols also. So we cannot use a matrix which is antagonistic to iron absorption. So this is not at all a good source of iron absorption. You should not be testing with gooseberry for improving absorption because it has almost 4 times higher amount of polyphenols. It's around 1 gram of polyphenol per 100 milligrams of gooseberry, but you can see the amount of polyphenols present in other fruits like Guava. It is only 300, and the other things are much less. So these are the other sources, but nothing like Guava and papaya or even the lime. But lime is again, the amount of ascorbic acid from 100 grams only around 48 milligrams, much less. So you probably need almost 2-3 limes a day to get the sufficient amount of improvement in absorption. Now, this is what has been suggested as strategy for Anemia Mukt Bharat, because they have given prophylactic iron and folate supplementation, then deworming, then in behavioral change communication, and also the test and treat process, and fortified foods, and also wash and related things. But nowhere there's a mention about a food-based approach, and I would probably would suggest popularize this food synergy to promote dietary diversification, and probably we should even test for vegetables not which with minimal processing to retain this one. Of course, there has to be efforts to increase production and consumption also. Avoid drinking tea. Now the next one, of course, the last portion is about fat soluble vitamins. Here the concept is again something very similar, because all fat soluble vitamins require some amount of fat for it to form micelles, because the absorption is through micellar processes, and both amount and type of fat both are important in absorption. And there are different steps involved in the micelle formation, like lipid matrix is very essential for the food carotenoids. Here we talk about, because plant sources provide only provitamin A carotenoids or beta carotene, that gets into this micelle, then that is being taken up, and then there will be dioxygenase, which actually cleave the beta carotene to form vitamin A. Now there are three studies which I wanted to. This is a study done by NIN long back, Dr. Mohan Ram Vinodini Reddy and group. There are three groups. This is rice with spinach with no fat, 5 gram fat and 10 gram fat. They did actually, this is done in children after supplementation, that is 6 weeks supplementation. Then they actually estimated the serum vitamin A post withdrawal of supplement. That is to show whether there is sufficient store, and it's an indicator of a storage form of iron, not iron, vitamin A, and if the levels are retained, you can say sufficient stores are there. So 5 and 10 grams actually shows higher amounts of vitamin A. So it is, now our guideline also shows minimum 5 grams of fat is required for this one. This is another study. Carotenoid bioavailability is higher from salads ingested with full fat than fat reduced salads. You can see as you increase the amount of fat, all forms of carotenoids are actually at a higher level. So for absorption, this is with the bottom line is no fat. This is with a 6 gram canola and 28 grams canola. So beta carotene absorption is, I think, the maximum with that. Similarly, food matrix with 10 grams of vegetable oil. This also shows the same thing. And also the best one is with the red palm oil. It's a source of vitamin A. So actually, there is a study done showing that the absorption is very good. This is the orange flesh sweet potato, which has high amount of beta carotene. And there is this one study which shows that with a sufficient amount of corn oil, the absorption is good. Then the final one is about the vitamin D. Of course, here there is, though it is not a food synergy, here only thing I wanted to say is the sunscreens, which actually prevent the sun to produce enough vitamin A will be vitamin D is reduced. Now you have two scenarios. One is without sunscreen and with sunscreen. Okay, so if you are exposing your only the face, then with the sunscreen actually you get minimal exposure and minimal conversion of formation of vitamin D3, and so use minimal amount of sunscreen, and also the time of exposure that is required, also the what is called the zenith angle. This is the zenith angle, and it should be between 11 a.m. to 3 p.m. This is the time you get maximum exposure time and maximum what you call the erythema dose required to produce vitamin D. And this is a systematic review to show that the oil medium is very essential for higher absorption compared to powder or ethanol. So if you are having vitamin D as a powder, I mean, maybe the absorption it will be less, or even the D2 form, the ergocalciferol, which is we have now the irradiated mushroom, which is supposed to be a source. And also for fortification, we suggested vitamin D2 as a source. So they all require this oil as a medium. Now to conclude, I would probably suggest these evidences is good enough to practice this and make it as a national mission to address at least vitamin A, vitamin D, and iron deficiency is in the country. Thank you very much. And I
Thank
wait
you,
for
sir.
the
It was
yeah,
really,
question.
yeah, it was an excellent session. We felt, it should not get over only because you are a reservoir of knowledge and so much based on your research. You are giving so many important tips. Even we were not sure about many of those things. So thank you once again. Now, since we have a short period, we will just, can we start taking questions, sir?
Oh, sure.
Can we start with question answers? Yeah.
Sure.
There's one question is that one you mentioned about that CNN survey. Which year was it done? Because the person is asking that as per the seasonal data collection, the intakes of vitamin A may vary. Because the person who's asking the question is saying that in summer there's a lot of mangoes are available, so your vitamin A intake can increase. So when was this survey what you had shown was conducted? Which year?
This is done in 2016 to 2017, 1 year. It's a national survey, so it is not, and the seasonal variation is taken care of in this survey actually.
Okay.
I mean,
Thank you.
because it's been part of the design.
Yeah. Now, one question, we all know that we keep on saying that jaggery is better than sugar because it has iron. Then iron vessels should be used. Now, how far is it true that using iron vessels, how much of iron really gets in, and out of that, how much gets absorbed? That's one thing. And jaggery these days, I have heard that they use some steel or some other kinds of equipments. And so from where does the iron come? So is there any, what's your take on this kind of about?
Yeah, I think this is a common question asked, and I have actually written a review article recently in NFI Bulletin, which clearly and highlight there is it's a contaminant iron, and also the solubility. Again, it has to require an acid medium at the gastric environment. Unless that is there, all contaminant iron is not going to get absorbed.
Right.
It is through the same, and it is not soluble once you are actually elemental iron also to come from a kadai, okay, cooking vessel. You require iron in acidic medium. Okay, so without ice, that's why, I mean, if you look at the literature, they add certain citric acid, many other things to create that environment to increase the solubility. So the messaging
that
is
again
not
should
right.
not be too much also, right?
Yeah.
They also say that immediately
there is lot of miscommunication about that. It is not just katai. You have to add certain acid medium to increase the solubility. It's actually leaching out. The leaching will be, iron doesn't come out in normal, what you call aqueous phase or the water. It requires acid medium, at least pH around 2 to 3.
Right. Then there's one question
Even
is that
even the your jaggery also same issue.
right.
It's all contaminant iron.
Right. There's another question is, sir, does iron and zinc absorption changes as per the age and the clinical conditions?
My study actually shows that the at least gender has no influence. It actually um, uh, age definitely it will have, because as age advances, especially in males, the iron stores will increase, then the absorption comes down. So
and
your
some clinical conditions also,
clinical
the absorption is affected.
anemia and deficiency itself increases absorption.
So then
Because
supplements can be taken, supplements, the absorption from the supplements.
Yeah, there will be better, but that is not enough, because the solubility of again is limited to the acid milieu in the gastric phase. It doesn't produce so much acid to solubilize 100 milligrams of iron.
Right. Now, one question, you have mentioned that the pearl millet has good amount of iron, and then iron millets also have phytates. So what about the bioavailability of iron from millets?
I showed one actually a study showing that it is only 4% absorption. So absorption is very, very poor.
Okay.
So unless you have the food synergy,
right,
and one has to test it whether you can increase the absorption, and it is it's iron rich, but absorption is very poor.
Right. There's one question, sir, how to help low-income group population to bring about diet diversity when minimum income they have? So which foods do you suggest they should have, which will bring the right kind of diversity and then the food synergy?
Yeah, definitely there should be an increase in the number of food within the green leafy vegetables or other vegetables and fruits that needs to be included. Now, some of these are green leafy vegetables locally available. Even Dr. Bamji was talking about that yesterday. Probably you should practice that Nutri garden or kitchen, which is part of your symposium also. I think there will be somebody talking about IYCF and Nutri kitchen gardening, right?
Yeah.
Yeah, so I think
No,
it has to be
sometimes.
a mission mode. This thing, I mean, there has to be a good messaging of this so that people practice it.
Sir, sometimes it is said that whatever those vegetables which are grown in villages, for which, the nutritive value is not available in our table, our books, IFCT and all. So how to go about, like if we want to promote consumption of those vegetables, but we don't know, maybe we may analyze some iron and some nutrients, but there could be some anti-nutritional factors or something, So how to go about, promoting consumption of this kind of vegetables?
If it's unconventional, of course, one needs to do an analysis, but the conventional foods, you don't find much variation in the content.
Yes.
Because whatever is reported in that IFCT, that is Indian Food Composition Table, they are average of from different regions.
Right.
It's not from
Then,
one region.
sir, suggestion has come, which of course we also talk about, is that the fermentation and then the soaking of millets, cereals can enhance the availability because the phytate content is reduced. So is it true, and to what extent it can be reduced? The absorption can be increased and the phytate and the,
Yeah, phytate has to be considerably reduced. So because we have actually when published one on the germination and how much of increase in bioavailability, it is not much unless you almost reduce the phytate content.
right.
Even because it depends upon the molar ratio of iron to phytate.
Right.
But it can be practiced, but we may not be able to get the desired effect in short time.
Right. Okay, sir, there are many more questions, but I'm very sorry because of time constraints, I won't able to take. I want to tell the participants that there are some questions are there and which we will be mailing it to the today's expert, Dr. Nair, as well as yesterday also some people had questions which could not be answered because of the time constraint. They will be sent to the expert, and as well as the answer will be sent back to the person who had asked the question. So we will just wind up this session now. So once again, sir, I take this opportunity on behalf of NSI Mumbai chapter to thank you for sparing your valuable time. And I think you have given so many wonderful tips, all based on your research. Now, something like Guava, I think like instead of saying apple a day keeps doctor away, we should say that Guava a day keeps anemia away, and try to incorporate everywhere. Even papaya, because apple and papaya is something which is, I've seen in rural areas, they are grown abundantly. And so, these tips are really important, and dietary diversity, as you said, is possible even with the minimum, the low income, where the kind of fruits that you have suggested, papaya and all that, even maybe red pumpkin, drumstick leaves or whatever. And so that is the need of the hour, as from your session, it is emerging that we need to be very, careful and then balance our diet, like what you have said right in the beginning. Balanced diet is very important, that if you have incorporate all various food groups, automatically everything is taken care of. So that's very nice. So sir, once again, thank you very much, and look forward to have many more sessions with you, of a little longer duration. Sorry, today we have to stop. So now with this, we have come to the end of the first session. Now may I request Mrs. Shama Chavan to take over the proceedings of the next session. So thank you very much.
Thank you very much. Wish you all the very best.
Thank you, sir. Thank you, Veena ma'am. Good afternoon to one and all. It's indeed an honor to introduce the second speaker for today's session, Dr. Sadhana Joshi. Next slide, please. Yes. Dr. Joshi's educational qualification and professional experience. She is a PhD in nutritional biochemistry from Agarkar Research Institute, Pune. She's joined Interactive Research School for Health Affairs, Bharati Vidyapeeth University, Pune as a senior scientist in 2004. Currently, she is a director grade scientist and heads the Division of Mother and Child Health at IRSHA. She has been working in the area of mother and child health for over 25 years. Next, please. Her achievements. Dr. Joshi has several novel findings to her credit, which are published in 140 journals, with the large number being published in international journals. The group has received a large number of grants from various departments of the Government of India, like the DBT, ICMR, and DST. In addition, there are a number of student projects, junior, senior research fellows, and research associate, which are funded by the ICMR, DST, and CSIR. She is also the principal investigator on an ICMR Center for Advanced Research, where she will examine the associations of maternal long-chain PUFA and micronutrients with clinical outcome in preeclampsia. Next, please. Her awards and achievements. She was bestowed on the 9th Dr. Rajamala P. Devadas Memorial Award at the 5th Annual International Conference of the Nutrition Society of India in 2018. Her research, she has received the Research Excellence Award for developing country scientists at the 7th World Congress on Developmental Origins of Health and Disease, Portland, USA. She has also received the Best Researcher Award at the 15th Foundation Day ceremony at Bharati Vidyapeeth University, Pune in April 2010. She is at the editorial board member of Journal of Nutrition and Intermediary Metabolism elsewhere. She is reviewer of various international journals with high impact factor. She is also the member of the task force at DBT, an expert reviewer for ePromise, DBT, ICMR, and DST SERV. Next, please. She has been nominated on the expert committee on wellness counseling formed by the state project director, Rashtriya Uchchatar Shiksha Abhiyaan, RUSA, Maharashtra in 2017. She is board of member of the Preterm Birth International Collaborative Prebic Australasian branch. She was invited to participate in 2016 Grand Challenges meeting sponsored by Bill and Melinda Gates Foundation at London. She has also been invited to deliver talks at several national and international conferences: World Congress BCGIP India, Annual Congress of Society of Fetal Medicine, Nutrition Society of India, Indian Dietetic Association, and a number of other national and international conferences. A total of 16 students have obtained their PhD degree under her guidance, and 6 others are currently pursuing their PhD under her able guidance. She is a life member of various national and international organizations. I request and also welcome Dr. Sadhna Joshi for taking on this session on role of preconceptional nutrition on pregnancy outcomes and future health evidence-based nutritional concepts. Over to you, Dr. Sadhna.
Sadhna ma'am, can we
Yeah.
request you to adjust your camera, please?
Yes, ma'am.
Yeah, is that
Yes.
better?
A little lower, ma'am, if possible. A little bit more lower.
Is that okay?
No, ma'am.
Yeah, ma'am,
Yes,
right now, yeah.
this is better.
Thank you,
Okay.
ma'am.
Yes.
Okay. Can I
Thank
share
you
the
so much.
screen now?
Yes, ma'am.
Yes, ma'am.
Okay. So good afternoon to all of you all. I would like to thank the organizers, the Mumbai chapter, for giving me an opportunity to kind of discuss the work that we have been doing over here. Actually, we are not pure nutritionists. We come from the biochemistry and the biotechnology background, most of my group members. So we would definitely value your comments also on our work. And I've been asked to talk on the role of preconceptional nutrition on pregnancy outcome and future health, evidence-based nutritional concepts. This is the work that is being carried out at the Interactive Research School for Health Affairs,
We are a constituent unit of the Bharati Vidyapeeth deemed to be University at Pune. So we all know that maternal nutrition plays a key role in determining pregnancy outcome. And as pregnancy advances, there is an increased nutritional need for macronutrients and micronutrients. And very purposefully, I have added over here the long chain polyunsaturated fatty acids, since most of my talk would be dedicated to the long chain PUFAs. Now, we also know that a poor maternal nutrition results in poor fetal growth, low birth weight, and higher infant morbidity as well as mortality. Now, these nutrients, I think you are all aware, play various roles during pregnancy, starting right from implantation. They're involved in placental invasion, angiogenesis, immune response, anti-inflammatory response, and they also regulate the synthesis of various hormones. I have now taken over here key micronutrients, which are part of the one carbon cycle and very relevant to the Indian population in determining the pregnancy outcome. The first one being folic acid. We all know that the Indian population is deficient in folic acid. Its deficiency results in neural tube defects, spina bifida, and the Down syndrome. In addition, they are also suggested to be important for oocyte quality and maturation. Vitamin B12 is widely discussed. Indians have a low vitamin B12 status, increasing the risk for spontaneous abortions as well as being associated with neural tube defects. In addition to these micronutrients, the polyunsaturated fatty acids consisting of the omega-3 fatty acids and the omega-6 fatty acids play a critical role in determining pregnancy outcome. These long-chain polyunsaturated fatty acids, they have various functions. They act as ligands for various transcription factors like PPAR, RXR, LXR, and they influence gene regulation. The omega-3 and omega-6 fatty acid pathway, they are either pro-inflammatory coming from the omega-6 fatty acids, that is the eicosanoids, and such as prostaglandins, thromboxanes, and the omega-3, which are anti-inflammatory like resolvins and protectants. And there has to be a balance between the pro-inflammatory and the anti-inflammatory metabolites, and whereby they are regulating inflammation. LC-POFAs are also involved in membrane synthesis and mainly maintain membrane fluidity, which helps in the intercellular signaling. Now, absence of these three vital nutrients results in poor fetal growth, is quite well established. And a poor fetal growth has also been shown to be association with non-communicable disease risk like hypertension, cardiovascular disease, and type 2 diabetes. And this concept was originally put forth by late Professor David Barker from Southampton University, where they suggested that the early environment during development is a strong predictor of phenotype and disease in later life. This concept currently is referred to as the developmental origin of health and disease. It states that during critical periods of development, nutrition can influence various developmental pathways and leads to permanent changes in metabolism and increases chronic disease susceptibility. So how does this do that? It is mainly through epigenetic modifications. Yesterday we heard Dr. Bamji say that epigenetics plays a key role, and this is where nutrition comes into the play over here. The epigenetics is influenced by various factors such as environment, but the main driver of epigenetics here is the nutrition that the mother consumes during pregnancy. Epigenetics was first described by Conrad Waddington in 1942, and it is a study of heritable changes in gene activity which are not caused by changes in the DNA sequence. This is an interaction between the genome and the phenotype, where it interacts with the environment such as temperature, radiation, nutrients, drugs that are used and which produce both the immediate as well as the long-term effects. And there are various mechanisms which help in this epigenetic transformations. They are mainly, Epigenetics can be either through DNA methylation or through histone modification. Now, DNA methylation is one of the most important and well-studied epigenetic mechanisms, which involves the addition of a methyl group to the fifth carbon of the cytosine base by DNA methyltransferase. And histone modifications are covalent modifications at the amino acids of the amino acid tails of histones, where they affect the overall chromatin structure. In addition to these two mechanisms, there are non-coding RNA, which are long and small RNA molecules which inhibit gene expression or translation by degrading the targeted mRNA molecules. Now, DNA methylation, as I mentioned to you, is very widely studied. There can be of two kinds. There can be global DNA methylation, which is methylation over the complete genome. Or they can be gene-specific methylation, meaning that there is a methylation over a very specific gene promoter. For example, angiogenesis during pregnancy is influenced by vascular endothelial growth factor, and it is very likely that this gene promoter could be methylated as a consequence of the maternal diet. And this methylation helps in either switching on or switching off of the genes concerned. That means when there is an active chromatin, the unmethylated cytosine residues are there and the acetylated histones, they help in switching on of the particular gene, resulting in increased gene expression. Similarly, they can either be switched off, and thereby they help in modifying the function of the genes and affecting the gene expression. Now, how do they do it and how is it relevant to during pregnancy? We all know what happens is once after fertilization, when the zygote is formed, there is de novo demethylation takes place. Genome-wide demethylation is there at the time when blastocyst is formed, at the time of the implantation. Once again, there is a genome-wide tissue-specific methylation which takes place. So if your maternal diet is inadequate, or if you are malnourished in particular nutrients like folate, B12, or the long-chain polyunsaturated fatty acids, it can affect methylation, and that can be dependent on the transition from the zygote to the fetus at the point at which the malnutrition occurs. So what is the main organ through which these epigenetic transformations take place? It is mainly through the placenta. It is considered that the placenta is the main organ which influences risk for diseases in later life through epigenetic modifications. We all know that the placenta is a temporary organ which anchors the conceptus to the uterine wall. The major function of the placenta is to transfer nutrients, gases, and hormones to the fetus. So what happens is that if there is a suboptimal maternal nutrition, it results in disruptions in the placental epigenetic profile, which in turn alters the gene expression. This together will result in aberrant placental development and result in adverse pregnancy outcomes such as preeclampsia, which is well known that it is a placental pathology. Similarly, gestational diabetes mellitus, we know that it results in a macrosomic baby. So this results, the aberrant placental development will result in kind of an IUGR or a macrosomic baby, and this is responsible for fetal programming, leading to chronic diseases in adult life. Now, coming back to the nutrients, the LC-PUFAs, we all know, are required during different reproductive phases, starting right from preconception. An adequate LC-PUFA status is required in the preconception phase so that pregnancy begins in a healthy state. During the first trimester of pregnancy, LC-PUFAs are involved in implantation and trophoblast invasion. During the second and the third trimester, the LC-PUFAs play a key role in the growth of the mammary glands, placenta, uterus, and fetus. And finally, they are also involved in placental development and maturation. Postnatally, they play a key role because these are the main lipids which are found in the brain. So they also influence the baby's nervous system and the cognitive functioning of the child. So you can see here that LC-PUFAs play a critical role in the overall fetal growth and development. Now, these LC-PUFAs, together with folate and vitamin B12, they form the part of the one-carbon cycle. Traditionally, the one-carbon cycle consists of folate and vitamin B12 and homocysteine. In addition, we all know that folate and B12 are major methyl group donors. And we have for the first time from our group shown that there are also methyl acceptors in the form of phospholipids, which are carriers of the LC2-phase. So what happens over here is the dietary folate interacts with vitamin B12 to form S-adenosyl methionine, which is a major methyl group donor. The methyl groups are donated to the acceptors, that is phospholipids, DNA, or histones. In the absence of these phospholipids, the DNA or the histone gets methylated, resulting in altered expression of various transcription factors, causing an adverse pregnancy outcome, and as I mentioned to you, may increase the risk for non-communicable diseases in later life. Now, there are different critical periods which can influence these epigenetic transformations. Firstly, I would like to begin by saying that the diet in preconception plays an important role in influence the long-term health of the offspring. Similarly, during the prenatal period, the Dutch hunger winter from 1944 to 45 is a very classic study which showed that the limited food intake of mothers who were pregnant during this period influenced the health of the children. These are all retrospective studies which were analyzed later on. Similarly, the postnatal phase is also important in determining the permanent effects in adulthood. Based on all this, we carried out a proof of principle study using an animal model where we had, you can see here, different groups: the control, the folic acid deficient, and the vitamin B12 deficients. And since the folic acid and vitamin B12 we have shown interacts in the one carbon cycle, we supplemented these two folic acid and B12 groups with omega-3 fatty acids. And here you can see the study began right from pre-pregnancy, that is around 16 weeks prior to pregnancy, and we monitored its effects on the estrous cycle. When after the animals were put for breeding, we examined its effects on pregnancy, the weight gain, litter size, after which the pup weight was followed up during lactation, and the pups were dissected postnatally at day 22. You will see over here that when the diet was deficient in either folic acid or vitamin B12, there was a significant decrease in the number of the estrous cycle, whereas supplementation was able to ameliorate it to some extent. Similarly, you all know that there are different phases of the estrous cycle. You can see here in the control diet, the phases completely are different as compared to the B12 deficient diet and the omega-3 fatty acids addition, although to the vitamin B12 partly could ameliorate, but it was not able to bring it back to normal as per the control animals. Similar study was also seen for the folic acid deficient group. You will see here that the folic acid deficiency altered the phases as compared to what you would see in a control animal. Then when we look at the weight gain, you can see over here that when the folic acid deficient animals, they had the lowest weight gain, and supplementation was able to improve it to some extent. Looking at the litter weight and the litter size, you will see here that there was a significant reduction in the litter weight in the vitamin B12 deficient animals, although such effect on a litter size was not seen. This deficiency of folic acid and vitamin B12 also influences various hormones like progesterone and the estradiol. Here, the progesterone levels were significantly higher in the deficient, both the micronutrient deficient groups, that is the folic acid as well as the vitamin B12. Similarly, the estradiol levels were also higher in both the groups. Then we also looked at the morphology of the mammary gland. You will see that in the case of a vitamin B12 deficient animal, there was an absence of the lactating duct, suggesting very clearly that B12 deficiency during the preconception and pregnancy period can influence lactogenesis in the animal. Similar picture was also seen for the folic acid deficiency, where there was an absence of the lactating duct. Then we also looked at the mammary glands. After the mammary gland, we also looked, you will see here that the corpus luteum was significantly decreased as compared to the control animal. Then we looked at the plasma malondialdehyde levels. Malondialdehyde is a marker for lipid peroxidation. You will see over here that the folic acid and vitamin B12 significantly increased the plasma malondialdehyde levels, and this was partly ameliorated by the omega-3 fatty acid supplementation to those groups. Similarly, the placental TNF-alpha levels, these are pro-inflammatory cytokines, that is TNF-alpha and interleukin-6, was also examined. Micronutrient deficiency increased the levels of pro-inflammatory cytokines in both folic acid and vitamin B12 deficient animals. Then, as I mentioned to you, we also looked into global methylation levels. So you will see over here, the micronutrient deficiency significantly increased the global methylation levels here, showing very clearly that maternal nutrition does influence epigenetic patterns in the liver of the offspring also, not only the mother, but even the offspring. So to summarize this work, I would like to say that a deficiency of folic acid or vitamin B12 resulted in an altered 1-carbon cycle, which altered the estrous cycle. It impaired the mammary gland and ovaries and reduced the fertility. On the other hand, it also had played a key role in lowering the placental docosahexaenoic acid levels, suggesting very clearly that the micronutrients interact with the LC-PUFAs. It reduced placental PPAR gamma levels and thereby placental development, resulting in an adverse pregnancy outcome. Similarly, we could also show that not only the mother, the offspring also showed lower levels of DHA, PPAR alpha and gamma, which are transcription factors, and it altered the liver fatty acid metabolism, maybe ultimately increasing the risk for non-communicable disease in later life. Now, in addition to this folic acid and vitamin B12, we heard Dr. Bamji also say yesterday that vitamin D deficiency is also seen over here. Though originally it was thought that Indians may not be deficient in vitamin B12 due to the abundant amount of sunlight which is present over here. But you will see that India is one of the countries which are most deficient in vitamin D. So this vitamin D also plays important roles during pregnancy. Vitamin D is involved in fertilization and immune regulation, plays a key role in calcium homeostasis, glucose homeostasis, muscle health. It regulates the blood pressure and ultimately improves cardiovascular health. In the placenta, it plays a key role in immunomodulation, anti-inflammatory responses. It is involved in extravillous trophoblast invasion, thereby regulating implantation, desudalization, and also regulates placental hormone production. In the fetus, it plays a key role in lung maturation and the neurodevelopment. So the next, we hypothesize here that both these long-chain polyunsaturated fatty acids and LC-PUFA together may be interacting to lead to a wide spectrum of adverse pregnancy outcomes such as preeclampsia, small for gestational age, or low birth weight babies. To study this, we carried out an animal model where during the preconception period, the animals were made deficient in vitamin D. And the control animal, of course, received the control American Institute of Nutrition guidelines. The diet was as per that. And here you can see we examined the whole range of parameters starting from long-chain polyunsaturated fatty acids, both in the plasma, liver, placenta. The oxidative stress marker, the ELISA was done for undertaking inflammatory markers, homocysteine, vitamin B12, folate, and we also examined the expression of a large number of genes which were involved in vitamin D and long-chain polyunsaturated fatty acid metabolism. So you will see here when the animals were made vitamin D deficient, there was a significant reduction in the total gain. The pregnancy weight gain was much lower as compared to the control animal. Similarly, the placental weight was also much lower as compared to the control animals. And the vitamin D deficiency also significantly influenced the fatty acid metabolism. As you can see that arachidonic acid, that is the omega-6 fatty acid, this is the last end product of the omega-6 pathway. It is a key molecule which was significantly higher. It leads to the inflammatory pathway. And you can see that the deficiency of vitamin D increase the arachidonic acid, and along with that also many of the saturated fatty acids like stearic acid was also much higher. Does that mean that the conversion of the omega-3 fatty acids from ALA to DHA was affected because of the vitamin D deficiency? And therefore, we looked at the delta 5 and delta 6 fatty acid desaturases, which are the key enzymes which regulate the conversion from the alpha-linolenic acid to the docosahexaenoic acid. And you can see here that very clearly this conversion is affected because of vitamin D deficiency. So I can summarize over here that we had earlier shown that folate and B12 deficiency increase homocysteine levels and also increase your oxidative stress. And this was interlinked with the fatty acid metabolism. Now, to add to that, we also find that the vitamin D also interacts in the 1-carbon cycle by regulating cystathione beta synthase, increasing the arachidonic acid, and thereby it creates an imbalance between the omega-3, omega-6 pathways, resulting in placental inflammation, endothelial dysfunction, and increasing the risk for various pregnancy complications. So to summarize this part, we can say that changes the intrauterine environment, especially the 1-carbon cycle in the mother, along with the low omega-3 fatty acid status, increases homocysteine, increases oxidative stress, alters placental epigenetic profile, and increases the risk for chronic diseases in later life. All these studies we also carried out on a large number of cross-sectional studies in humans, where a similar kind of pattern was seen. And we know that now I all have been mentioning that there are two kinds, omega-3 and omega-6 fatty acids. Now, just to kind of brief up, the omega-3 fatty acids, the precursor is the alpha-linolenic acid. The source is dark green leafy vegetables, flax seeds, walnuts, kidney beans, canola oil, and soybean oil. But the main end product here is the docosahexaenoic acid, which is mainly found in fatty fish and fish oils. Similarly, the omega-6 fatty acids are found in abundance, and their sources are sunflower, safflower, cottonseed, and soybean, and meat and eggs. Now, as I showed you in the animal study, we showed the significant beneficial effects of the omega-3 fatty acids, where mainly the supplementation was the docosahexaenoic acid was used along with the eicosapentaenoic acid, both of which are mainly found in non-vegetarian sources. So this raises a question that what happens to an Indian population which is 30% vegetarian in nature? The overall fish intake in India is also very low based on the NNMB report. And we also know that the conversion from the alpha-linolenic acid to the docosahexaenoic acid is very poor. It ranges from 0.5 to 9%. But most of these studies were carried out on Western individuals and who possibly are not habituated to consuming high amounts of alpha-linolenic acid in the form of green leafy vegetables. And studies have also shown that there's a positive association of maternal DHA status with birth weight. Now, we don't know whether the dietary supplement, if you give in the form of green leafy vegetables, we heard yesterday lots that green leafy vegetable consumption, Dr. Madhavan Nair also mentioned just now, can have significant beneficial effects. Now, the question here we raised was, will a green leafy vegetable consumption in any form improve the docosahexaenoic acid status in a non-pregnant mother? And here we hypothesize that a micronutrient-rich dietary supplementation, so it was made in the form of a snack, it was made from green leafy vegetables, fruits, and Mumbai, for 12 weeks to women of reproductive age from urban slums, will improve the fatty acid profile and reduce oxidative stress. Now, this work was carried out by one of my PhD students in collaboration with the Southampton University, and the study was carried out at Mumbai. So you will see here that there was a increase consumption of this green leafy vegetable, increase the docosahexaenoic acid consumption marginally. You can see there was a significant increase in both. It also increased the total omega-3 fatty acid after 12 weeks of consumption of the supplement. So we can say that a daily consumption of a snack containing green leafy vegetables, fruit and milk improved women's erythrocyte levels and without increasing the oxidative stress levels, because it is also likely that increased consumption in the form of ALA has been shown to increase the oxidative stress. But in this particular study, when it was given as a combination of green leafy vegetables, fruit and milk, there was no such increase in the oxidative stress levels. However, a lot more of work needs to be done to take this ahead. This was just a small study which was carried out by a PhD student. So based on all this, we at IRSHA have received a ICMR Center for Advanced Research. This study will mainly examine, it's a prospective study where we are recruiting women right from early pregnancy, from the 11th week of gestation, and following them up longitudinally for across various time points where blood is collected, the placental samples are collected, cord blood is collected, and the children are followed up. So this is a prospective study which examines the association of maternal long-chain polyunsaturated fatty acids along with the one-carbon metabolites such as folic acid, vitamin B12, and all the various biochemical measures which I mentioned to you with fetal growth in preeclampsia. And here what we are trying to do is we are trying to understand the biochemical and molecular mechanisms underlying this risk for preeclampsia. This preeclampsia is a pregnancy disorder which is characterized by high blood pressure and invariably leads to an IUGR baby. So in addition to this, there is also another early intervention study where this is also referred to as an Einstein, that is early intervention to support trajectories for healthy life in India. Now, till now, we have heard that we all know that preconception nutrition plays an important role in determining pregnancy outcome. So this study is actually coordinated by the World Health Organization, and this is a multifaceted intervention package to improve child growth and development. There are various international cohorts which are involved in this initiative, and that is mainly partnered with countries where there is a rapid rise in non-communicable disease and where they are trying to develop international intervention cohorts, mainly focusing on risk factors for non-communicable diseases. And all these are based on the concept of DOHAR. You will find over here that the major collaborators are DBT India, that is the Department of Biotechnology India is driving this study in India over here, the CHIR from Canada, South Africa, MRC, as well as China. And the whole study is, it's a very huge study for 10 years, which is coordinated by the World Health Organization. So you will find that there are a lot number of different collaborators in India where we also are a part of it. And the whole study is being conducted at Mysore, that is the Holsworth Memorial Hospital. And this is undertaken by the Swami Vivekananda Youth Movement over here. That is, it is undertaken in 12 different villages which are near to Mysore. And there are various groups which would be examining various different aspects of the study. We at IRSHA are mainly involved in understanding the various biochemical mechanisms of the study. Now, as I mentioned to you, this is a multifaceted intervention program which starts preconceptionally and continues through pregnancy and infancy, and it comprises measures to improve maternal and infant nutrition, which would help reduce the burden of non-communicable disease. The best part about this whole program is that it helps you tease out whether intervention starting from early conception is required, or if you start during pregnancy is good enough. There's always a thought that yes, you need to start it much before the preconception, but there were no studies which very systematically looked into this. So this has two different arms, one where the intervention is started right from preconception and continues throughout pregnancy, another arm where the intervention is undertaken only during pregnancy, and of course the control arm. So this study is already 2 years, it has been progressing. So once we hear more about the outcome of the study, things would be more clear. The main objectives, among the various objectives, is to assess the efficacy or the effectiveness of an integrated longitudinal intervention program to improve child growth, body composition, neurodevelopmental as well as cardiometabolic outcomes in the offspring. And of course, it builds a huge biorepository for undertaking future mechanistic studies. Now, just to briefly describe to you here, the intervention, they are not only nutritional. Of course, nutrition is a major part of it, where the anthropometry blood will be taken, but it also involves understanding the mental health of the woman, the cardiometabolic, physical activity, blood pressure of the mother, and the micronutrient supplementation here is as per the UNIMAP. And it also, along with the nutrition supplement, it also talks about lifestyle behavior change, that is healthy conversation skills by various community health workers, where they try and interact with the pregnant woman and try to modify her lifestyle patterns. During pregnancy, once again, there are community-based group parenting programs which are integrated with cognitive behavior therapy to address perinatal depression and improve the mother-child bonding as well as the child development, and to also include breastfeeding support. Postnatally, there are introduction of infant weaning foods, use of safe water for infant feeds, full vaccinations, etc. are the part of the intervention strategy. So I would like to end my talk by saying that definitely early interventions will help reduce the risk for non-communicable diseases. More such integrated approaches for non-communicable disease and mother and child health programs are needed. Healthy is one of the major programs which is being undertaken by the World Health Organization. And maybe this together, when you do, will help to help ride the rapid rise of non-communicable diseases, which is currently being witnessed around the world, especially in India, which is said to be the leader of diabetes in the near future, and high rates of cardiovascular risk is there. It may help to prevent some of this rise that we are seeing over here. If we are able to achieve that, we may be able to change the current scenario where we know that every 10 minutes a woman dies in India from pregnancy complications such as preeclampsia during childbirth. India also tops the list of countries with the highest number of child deaths, nothing that we should be proud about, but this is the fact. Similarly, the non-communicable diseases are also increasing at an alarming rate in India. So definitely it calls for studies which to address these issues right from the preconception stage. If we are able to do that, yesterday we heard Dr. Bamji mention about the Millennium Development Goals. We are nowhere close to achieving the Millennium Development Goals, and the crucial factors which influence that you can see over here is maternal nutrition. The Millennium Development Goal 4 says that there is a need to reduce child mortality, and 5 is to improve maternal health. So if we are able to undertake such kind of studies, we would go a long way in probably trying to achieve these Millennium Development Goals, to which India was a signatory in 2000. So this is my group. A lot of my students, we have a very large group consisting of over 45 PhD students, staff members are there, technical staff is also there. So all this work, I am only here to just present this, is majorly done by all these girls over here. And I would like to say it's an all-girl group which has played a key role in bringing our work till here to date. So once again, I would like to thank the Nutrition Society of India, Mumbai chapter, for allowing us to share our work with you all. So thank you very much.
Thank you, ma'am.
Yeah.
Thanks a lot. With your kind permission, maybe take few questions. Hello. Ma'am, can we take few questions?
Yeah, sure.
Thank you, ma'am. Ma'am, the first question is, in the light of deficiency of LC-PUFA generally seen in the population in India, are the requirements of the same increased during pregnancy? And if yes, to what extent?
See, in India, there is not much information about the LC-PUFAs, but what is recommended is normally it is the omega-3 fatty acids such as docosahexaenoic acid and the eicosapentaenoic acid, together of which is 500 milligrams per day. But however, as I mentioned to you, our main source in a vegetarian population is alpha-linolenic acid.
Right.
where there are very few studies. So one of the reasons for this ICMR study is to understand what are the baseline levels in the population.
Okay, thank you, ma'am. Would the effect of omega-3 fatty acid from vegetarian sources be the same as that obtained from fish?
Studies, if you look up the literature, meta-analysis and all suggest that no, it will not have a significant
Same.
effect, because the conversion from alpha-linolenic acid to docosahexaenoic acid, as I mentioned to you, is very small. However, we are a population who are habituated to consuming a vegetarian diet,
Correct.
which is rich maybe in green leafy vegetables. So we do need a lot of studies to evaluate the effect of this daily turnover consumption of ALA to, even though it may be a small amount of conversion from ALA to DHA, but does it have any kind of a cumulative effect? That needs to be seen.
Ma'am, continued to this question only is that is the conversion improved during pregnancy?
No, there are no such studies which
Studies
show
which
whether
prove
it
that.
is no, because most of
Improved
the
or not?
studies using isotope studies have been carried out on non-pregnant women or
Okay.
men they have used, where they have shown there's limited conversion.
Okay, thank you, ma'am. Ma'am, another question is, as folic acid and B12 affects the lactogenesis at cellular level, how
Yeah.
early preconception supplements with B12 and folic acid should be started?
See, one going by literature, one would suggest that much before you become pregnant, you have to start taking care of your body.
Your
And it is
source,
not necessarily only folic acid or vitamin B12. It could
B12.
also be vitamin D. So as Dr.
Correct.
Nair mentioned, you need a holistic diet,
Balance.
which, yes,
Correct.
which
A
includes
balance.
everything. Yeah. So it
Right.
doesn't mean only the micronutrients. You also need to
True.
take care of the various macronutrients along with the
So the
micronutrients.
foods from all the 5 food groups have to be
Yes. Yeah.
included
What he
in.
just mentioned
True.
definitely has a major role in this.
Right. Next question is, what could be the public health measures to overcome the dietary deficiency of folic acid, which is very commonly seen in Indian population?
So I think
This,
already a lot of work has been going on where they say, yes, consume foods which are rich in this folic acid. But the flip side of it also is
Mhm.
that when we are giving the supplementation, there is a folic acid supplementation program antenatally where you will see that the folic acid supplementation is given. And if it is given on a vitamin B12 deficient diet, I
Yeah.
haven't shown you the animal model that we replicated over there, where
Okay.
we say we have abundant of folic acid supplementation in the absence of vitamin B12, it causes all the adverse effects. So when you
Right.
are giving any kind of a supplementation, it has to make sure that it is balanced. And when
Mm-hmm.
that means balanced in all the micronutrients which are interacting in the one carbon cycle, or
Correct.
even the other macronutrients also.
Right. Thank you, ma'am.
Thank
For
you.
a good pregnancy outcome, dietary LC-PUFA and nutrient per se supplementation could be encouraged or not?
Definitely, yes,
Yes.
you should. Yes, one should look into that.
Another question is, do you suggest any specific preventive approach for the group in urban population where more incidence of preterm and IUGR is observed?
So there, once again, I would like to say your preconception nutrition should be good. Your diet during pregnancy, of course, should be very good. You can have physical activity, appropriate amount of physical activity should also be present, because we recently undertook a study in the urban population where we
Okay.
find there is, in the higher socioeconomic population, we
Right.
have absolutely no physical activity at all, which
Correct.
is possibly also, we are analyzing
One
that
of the
data, but
reasons.
that is also associated with an pregnancy complication like preeclampsia.
Correct. Thank you, ma'am. I think there is a question related to the study which you have discussed. What was the composition of snack, milk, fruit, and spinach that was used in the?
Yeah, because we had different kinds of snacks which were given over here. So it
Hmm,
was made sure that it was equal in both the control as well as the treatment bar which was given. So it was made in the forms of maybe parathas, and where it was not there in the control, green leafy vegetables were added for the paratha. It was not there in the control diet. So there were a lot many recipes which were given to avoid boredom. So if someone is
okay.
really interested, we can share the paper which is published on that.
Yeah, sure, ma'am. Thank
Yeah,
you so
sure.
much. Ma'am, on behalf of NSI Mumbai chapter, I would thank you, and we are really grateful to you for a wonderful session in which you touched right from epigenetics to one carbon cycle and how the micronutrient have a effect on pregnancy outcome, and also how vitamin D can affect the along with LC-PUFA the outcome of pregnancy. Thank you very much, ma'am, on behalf of NSI. Thank you very much.
Thank you. Thanks a lot.
May I now call upon Dr. Neha Sanwalka to please take us through the session.
Yeah, thank you so much, Dr. Nair and Dr. Joshi, for the amazing, enriching first morning session. I request all participants to join us sharp at 3. We will start our next session, which is the third session for the webinar. So join us by 2:50 using the same Zoom link which has been sent to you. And we will meet again at 2:50 and we'll start the session sharp at